In this project, I will be teaching you about enzyme replacement therapies and rare diseases. We are made up of cells, and inside each of our cells are tiny structures called organelles. These organelles within the cell include the nucleus, the Golgi apparatus, the endoplasmic reticulum, or ER, the mitochondria, the ribosomes, and lysosomes, which will be the focus of my video. The organelles all work together as a factory. Each organelle has its own job, and they all work together in allowing the cell to function normally. Lysosomes act as the recycling bin and/or trash can of the cell. They use proteins called enzymes to destroy things in the cell that the cell does not need anymore. These enzymes also break down and reuse molecules much like a recycling bin. Within the cells, lysosomes, and other organelles, there are enzymes. Enzymes catalyze reactions, can break down compounds, and can move things within the cells. One of these compounds is glycogen. Enzymes catalyze reactions by allowing substrates to bind to their active site. The enzymes then aid the two substrates in undergoing the reaction. Once the reaction has occurred, the product is released. Enzymes in lysosomal storage disorders are non-functioning, which means they are unable to move or break down the compounds that are in the lysosomes. This causes a buildup of these structures in the lysosome. There are many lysosomal storage disorders, including Pompe disease, Gaucher disease, and Fabry disease, and their treatments are all enzyme replacement therapies. These are rare diseases and only occur in about 1 in every 100,000 people. To get a better idea and understanding of lysosomal storage disorders, I will be explaining Pompe disease. Pompe disease involves the buildup of glycogen in the lysosomes of the muscles. This buildup is due to a lack of function in the enzyme called alpha-galactosidase, which does not function properly in patients with Pompe disease and therefore cannot break down the glycogen stores in the lysosomes. This buildup of glycogen causes the muscles to lose function and to become weak. The injected enzyme gives the patient's body the enzyme it needs, and then the glycogen can be broken down and the muscle can regain a more normal function. Enzyme replacement therapies are made by harvesting the specific enzyme needed for the disease from Chinese hamster ovary cells. These cells are grown in bioreactors. Bioreactors are large metal basins that vary in size. They are sanitary and allow for the control of pressure, temperature, and food supply for the cells, without exposing the contents to contamination. Every bioreactor is filled with a combination of cells and the food supply for those cells. Inside of these bioreactors, the cells go through cell division and divide over and over again until the bioreactor is full. Then, through various levels of filtration and protein purification, the enzyme of interest is isolated from the other components of the cell. Enzyme replacement therapies are administered intravenously, which means through injection. The enzyme replacement therapy then supplies the body with the necessary enzyme, allowing the body's cells to regain a more normal function, as we saw with Pompe disease. As the patient's body is able to regain a more normal function, they have an increase in life expectancy and quality of life. Thank you for watching.